Be in your way. I can move. I can move. All right. Bookkeeping things. I think the two labs are maybe slightly off schedule in kind of how we're going through things. If you're in my lab, I just got around to sending you your workshop questions today. So we're going to put workshop off until November 2nd. Tomorrow we will meet in lab together as a group because we do have stuff that we need to go through. But don't worry about staring down the barrel of workshop. I sent you all an email that has your questions for workshop on them and assigned. Some of you are working on your own, some of you are working in pairs, and then you got the assignment last week in lab, but these are the actual questions that I want you to be working on. In the event that you are having trouble finding information, things like that, you can always come and talk to Dr. Judd or I. If you're in my lab, probably easier to talk to me. If you're in Dr. Judd's lab, probably easier to just talk to him, although he does know quite a lot about lichens as well. So those of you in the lichen group, whose name I forget because I think I left your experimental design sheet up in lab, but he's worked with lichens in the past. Um, so that's that. Blah, 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 blah. What's your plan for lab tomorrow? Are you meeting with individual groups or are you meeting as a class?
Individual groups. You guys
Okay, all right. Okay. I didn't grade your papers over the weekend. I know Dr. Judd didn't either, so we'll be working on those and getting those back to you as soon as we can. We are starting a new case study today. So this case study is centered on Kanza Prairie, which is this area of prairie just south of Manhattan, Kansas. So if you drive a couple of hours west of here on I-70, when you get to Highway 177, which goes up to Manhattan, that stretch of land to the northwest of the highway at that intersection is 8,300 and some odd acres of prairie that is overseen by Kansas State, the Nature Conservancy. It's a large-scale experiment that we will be studying for the next 3 weeks. At the end of this, there is a paper that we will read, Veen et al. It's a paper that was published a number of years ago about the outcome of this large field experiment that they're running there. And just like we took you through the statistics that they used in the Hendry et al. Paper, today we're going to take you through the statistics that they use in the Veen et al. Paper so that when you get to the methods, you kind of know what it is that they're talking about. Keep in mind that we're not going to require that on a test you do an analysis of variance, but we want to take you through how an analysis of variance is done so that you understand kind of what the outcome of that test is. And so that's what we're going to be doing today. Can you guys see that, or is there a glare on it? Okay, you can see it. Okay, good. Because I'm going to probably want to write on things on the board, and this makes it a little easier. Any questions about anything?
Don't forget to even look at the daily schedule to see when we're going to cover methods or we're going to cover concepts. And when the literature summary is due in November.
Yeah, the article should already be up there. I would advise that you start reading it now rather than putting it off, simply because the more times you revisit it, the more likely you are to understand it. And that's the first step to writing a good summary, is actually understanding what the paper is about. So, in the paper, they use this technique called the analysis of variance. This is one of the most common techniques used, statistical techniques used in evolution, ecology, cell molecular biology, etc. It's also used in the social sciences, in psychology. So for that reason, we go through this in a little more detail than we did with the principal component analysis, because principal component analysis, you can go your whole career and never have to use that. You're going to see ANOVAs being used pretty frequently, so it's a good technique to be reasonably well versed in. So, imagine And keep in mind, this is an example from a statistics textbook, and it's the dumbest example I've ever seen. Get past the stupidity of the example, but these people were interested in how fast plants would grow when you give them different types of water. This is actually the kind of experiment that students used to propose to do in this class, and we have over the years managed to steer you guys away from these types of experiments to better experiments. So in this case, there's a control. There's water with 2% glucose added to it, 2% fructose added, 1% glucose and 1% fructose, and 2% sucrose. So I guess the fundamental ideas behind this is that plants need sugar to grow, and so if you give them sugar in their water, then they will grow more. And so what they've done is they've created a series of 2% solutions. In this case, this is a simple sugar, a single ring sugar, if you've studied sugar structure in a chemistry class. Fructose is a simple sugar. It's a single ringed sugar. Glucose and fructose at 1% makes up a 2% solution, but it's made up of two simple sugars. But then sucrose is actually these two simple sugars chemically bonded to one another. And so this is basically a compound sugar, but it's a compound sugar made up of glucose and fructose. That's what sucrose table sugar is. So that's kind of the logic behind this. And then they measured how much these plants grew. And so this is growth increment. And this is how it is actually presented. Essentially, I imagine that the scientists measured the height of the plant at time 1, and then they measured the height of the plant at time 2, subtracted the height at time 1 from the height at plant 2, looked at that growth that they made during that time period, and recorded that. And for each of these treatments, they had 10 plants that they subjected to this plant torture. So these are the data. So if you were interested in knowing if there were differences in these data, what are some of the things that you could do to these data from the get-go without any knowledge of an analysis of variance? Okay, so you could all calculate an average. And why can I not spell today? Oh yeah, it's just a normal day for Dr. Plowinski. It's a Monday. It's a Monday after fall break. Did you guys have a good fall break? Except for Lottie, who had to come back.
It was great until I had to come back.
Until you had to come back. What else could you do with these data? So you could calculate an average.
Make a graph.
Oh, you could make a graph. What would that graph look like, do you think?
Probably like a dot, and then do a trend zone.
Oh, did you look forward to the next slide? Oh, okay. It makes me think that you looked forward to the next slide. So you could make a graph. And you could plot what the actual values are. This is growth increment on the x-axis. These are the treatments on the y-axis. Note how I'm talking about figures. I put a figure up there. What's the first thing that I do? I tell you what the x-axis is, and then I tell you what the y-axis is. I was just talking to one of my senior students who has to make a presentation later on today about this. There is a certain way in which you work people through a graph. Identify for them what the axes are, and then talk about what the data are, and then what the data mean. So in this case, we have growth increment on the x-axis, the treatments from control, simple sugar, two simple sugars together, and two simple sugars in the form of a compound sugar. These are the actual data points for the control. These are the data points for 2% glucose, 2% fructose, 1% glucose, 1% fructose, and 2% sucrose. How is this more informative or less informative than just calculating the mean and plotting the mean? You could have just plotted the mean.
It allows you to compare each result
Okay, but so would calculating the averages of these treatments, because the averages would just look. You could just plot the average. The average is here, the average is here, the average is here, the average is here, the average is here. What does plotting the individual points gain you or not? What does ANOVA stand for? Analysis of variance. What does this plot tell you something about that the mean does not? Tells you something about variance. How variable are the data? Which of these treatments gives you the most variable data? The control. The control is most variable. It goes from 65 all the way up to 76. A lot of variation. Less variation, less variation, less variation, less variation than the control. The rest of those treatments have similar levels of variation. What do you remember from our treatment of statistical hypothesis testing way back at the very beginning of the semester when we talked about how we can tell whether one mean is different from another mean? Is just knowing the mean sufficient, or do you need to know something else? What's that? You need to know some measure of variation. What's the standard measure of variation that we're going to be using typically? Is it the sample variance? Is it the standard deviation? The sample standard deviation? Is it the standard error of the mean? Is it the 95% confidence interval of the mean? Which one of these would you use for decision making about whether or not two means were similar to one another or not?
Sample variance.
Not sample variance. Thank you for playing. 95% confidence interval. Why? Somebody else. Don't use the word confident. You guys are in the same group, right? Yeah. Oops, sorry. And somebody else. Oh, Sadie. Oh, sorry. I'm stumbling all over the place. Sadie. Somebody else. Confidence interval. What's the meaning of a confidence interval? We gave you a couple of ways of defining it. Yeah.
I think in this class you guys are defining it as 95% of the data is invalid.
Almost. Not quite, though. But you spoke up earlier and you just spoke up now, so give yourself a point. He's close. What is he getting wrong? It's okay to be wrong, but it's better to be right. So let's be right. What's he missing? Is it 95% of the data fall within that range? Oh, you think I'm going to give you another point for being right now? All right, go ahead.
But it's 95% of the
95% of the sample means will fall within that range. So it's based on the distribution of sample means. If you go to that population and sample it repeatedly, sample it 1,000 times, each time you sample it for 10 plants that have been tortured for their growth increment, take those averages, you now have 1,000 averages, 950 of those averages will fall within the 95% confidence interval of the mean. So why is it important to distinguish that it's a 95% confidence interval of the mean? What are we interested in knowing is different here?
Variances.
Nope, not the variances. Well, actually, yeah, that is correct, but it's also not correct. What we're doing actually is we're using variances, various types of variances, to tell us something about what we really want to know about, which is, and for that we're going to measure what? The sample mean. We want to know if the means are different, so we use something that is based on the distribution of the sample means. So if we want to know the analysis of means, why do we calculate an analysis of variance? Well, it's because there are different kinds of variance. The type of variance that we have taught you about up to this point is within sample variance. You have a sample, you have a mean, and you look at how the individual values vary from that mean. So in the case of the control here, the average value turns out to be 70, and we calculate the within sample variance by asking ourselves, how far away from the sample mean is this value? Well, it's 0 units away. How far is this value away? How far is this value away? How far is this value away? And how far is this value away? On the high side, and then we also ask, how far is this value away? How far are these 3 values away from the mean? And how far is this value away from the mean? Now, if we add up all those differences, what's it going to sum to? Oh, I don't know. It's not going to sum to 70. So this is a difference of plus 1. Here are 2 differences of plus 5 and plus 5. And this is a difference of plus 6. This is a difference of minus 2, minus 3, minus 3, minus 3, and minus 5. 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 1, 6, 12. Whoa, that's not right. Oh, there's that one in the middle. I chose one that was split. Crud. What did I do wrong? 70. 1, 2, 3, 4, 5. That's 16. 17. I wonder if I have my mean in the wrong spot. Maybe I graphed my mean in the wrong spot. Generally, though, what should be on the plus side and what should be on the minus side? If it's centered exactly at the mean. It should sum up to 0. Who said that? Oh, okay. Great, Sonia. It should sum up to be 0. And so what do we have to do with these deviations from the mean, from the sample mean? When we calculate sample variance, do you guys remember what the equation is? It's the sum of the deviations of each individual variable minus the sample mean, but then what do we do with that? This would sum to 0, so what do we have to do to make it not sum to 0? Nope. Because then every variance would be 1, and that's not the case. Square it, because when we square it, the negative values get turned into positive values. We square that, and then we divide that by this number of degrees of freedom. So for each of these, wherever the mean is, we can calculate the within sample variation by taking the deviations from the mean, and when we take the deviations from the mean, we square them all, we add up all those values, and that tells us something about how far apart these values are from the mean. This thing up here is the sum of the squared deviations from the sample mean. I'm going to repeat that for you because it's crucially important. It is the sum, that's what the sigma is, of the squared, that's what the square is, deviations from the sample mean. This is a deviation of the first data point from the sample mean. Can also be a deviation of the second data point from the sample mean, the third data point from the sample mean, all the way up to the 10th data point from the sample mean. Each of those deviations is squared and then summed up. This is sometimes also referred to as the sums of squares, which is just shorthand for this, and it's oftentimes denoted SS for the sums of squares. Now I realize you're asking yourself, why is he telling us this? Because you're going to see a table later on, and one of the columns in the table is going to say SS. And when it says SS, it's referring to the numerator of a variance estimate. Because this is an analysis of variance. And one type of variance that you can have is within sample variance. There's within sample variance within control, there's within sample variance within glucose, there's within sample variance within fructose, within sample variance within glucose fructose, and within sample variance within sucrose treatment. Are there other kinds of variance that occur in this data set other than within sample variance? What other kinds of variance might there be? It's actually the variance that we're most interested in, right? Do we really care how much variance there is within samples in the end? What's more important to us? Who was that? Lottie? What do you mean by overall?
Like all of them together, the average.
So variance among what? Nope, that's what we did here. This is variation within samples. That's variation among individuals. Well, these aren't species. These are all like bean plants that were given different types of water. Variation among what? What's the y-axis here? Variation among treatments. So we gave them different kinds of water. I don't have enough pins to throw around. Lottie, point for your group. Oh, your group is up here. Sonia, can I fling it back there? Just keep that back there. Sorry, is it almost dead? All right, just toss it to the side. Next time I'll throw you a new one. All right. So we're really interested in the variance among the treatments, right? We need to know how much variation there is within treatments because within treatment variation can obscure variation among treatments. So how do we go about calculating among treatment variation? Well, what we do is we calculate this thing called the grand mean. Oh, crud. I messed myself up because I didn't know what was coming next. You guys know how to do a t-test? Have you guys done a t-test back in high school or something? How many of you have done a t-test? What does a t-test do? Okay. Somebody else who's done one who can remember what you do with the t-test. A t-test compares two samples. So one of the things we could do is we could ask ourselves, is control different from sucrose? That we would do that with a t-test. We could ask if control is different from glucose, if control is different from fructose, if control is different from sucrose. Then we could do, is sucrose different from glucose, different from fructose, different from the 1-to-1? We could then ask, is glucose different from fructose? Is glucose different from the 1-to-1? And we could ask if fructose is different from the 1-to-1. And we could do these comparisons through a bunch of t-tests. Well, the problem with that is that the more t-tests you do, the more probabilities you have of creating a type 1 error. Have you guys heard of type 1 error before? So a type 1 error in its simplest form is the probability, because we're talking about means here, the probability of saying that two means are different when they are actually not. So go back to the very beginning when I was talking about the distribution of sample means. So we have a population of plants given pure water. Some of these plants grow a little, some of these plants grow a lot, but most of these plants do what? They grow some intermediate amount. So we have a normal distribution of plant growth, where this is growth increment. Now what we do is we go to this population of all the bean plants, all the pea plants that are out there, we give them, we select 10 plants out of this population and we give them pure water. So now we have a sample of 10 plants, and from that we can calculate the mean for the control, right? So now, because we have nothing but time on our hands and an unlimited amount of cash, we now do that a second time. We get another second of 10 plants, and we have control, and there's a second measure of the control average. We do that a third time, we get a third control, and we do that 1,000 times, and then we can create a distribution of sample means. Now, this distribution of sample means has a mean that is close to the population mean, but we can look and see the range of values within which 95% of all these sample means falls. That's the 95% confidence interval of the mean that we would have calculated on the basis of the first sample. That 95% confidence interval of this sample mean should include 95% of the sample means that we draw out of this initial population. Does it include all of them? No. There's 2.5% of them that are out here, and there's 2.5% of them that are out here, right? So if we then go and get another mean, and this is now the mean for the 2% glucose treatment, that mean can either fall within this range. If it falls within this range, what would we conclude about whether or not the glucose treatment is different from the control? If it falls within the range, we would say that they're similar. They're not different because we could have easily drawn that average growth increment from this distribution of sample means. If it falls outside of that range, what would we conclude about the difference between glucose and the control? That it's different. Now, what if it falls outside of this range, but it falls right there? Would you still conclude that it is different? Well, if we're basing our decision on whether or not means are different or not on the 95% confidence interval, would we say that it's different or would we not? Emma?
You say that it is different because 95% of it is supposed to go within there.
Yeah, we would still conclude that it's different because it's outside the 95% confidence interval. Now, could it have come from that distribution of sample means? Oh yeah, it's possible. Is it likely? No. So when we say that we're calculating a 95% confidence interval, we're basically saying that we want to define that distribution of sample means in a way that anything outside the bounds of that confidence interval is really unlikely to occur if those two means were drawn from the same population. And so we calculate the 95% confidence interval because we've chosen what is called the alpha of 0.05. Notice that 1 minus 0.05 is 0.95. That's where the 95% confidence interval is. The alpha basically says that when we think about the distribution of sample means, we want 5% of the sample means to be outside the bounds of our 95% confidence interval. Alpha is the type 1 error rate. And so what it says is that when you see two means and you compare them using 95% confidence intervals and you conclude that they are different, there is a chance that you're saying two means are different when those two means could have come from the same distribution of sample means. In that case, you've made an error. You said two means were different when in actuality they could have come from the same underlying population or the same distribution of sample means. That's why it's called type 1 error, because you have made an error of concluding that two means are different when in actuality they're the same. If you conduct a lifetime of experiments, you have made some incorrect conclusions because we are almost always working with an alpha of 0.05 or an alpha of 0.01. And so there's always a chance that you're concluding that two things are different when in actuality they're not. That's what a type 1 error is. Now, if we do these multiple t-tests, there's 10 of them for this data set. Each one of these has an alpha level of 0.05. If we do all of those tests, what is our type 1 alpha level for all of those tests? It's the sum total of all of those alpha levels. So in this case, what would the alpha level for the entire experiment be? 0.05 times. So now, the chance that you're going to say that two of those are different among all of those combinations is 50%, saying that they're different when in actuality they're not. If you're going to work with an alpha level that is that large, you might as well just not do the experiment and do what? Flip a coin. Is mean A different from mean B? If it's heads, it's different. If it's tails, it's not. Is A different from C? Flip a coin. If it's heads, it's different. If it's tails, it's not. You would get the same result from flipping a coin. Well, if that's the result that you would get in terms of your ability to distinguish means from one another, there's really no point in doing the experiment. So we needed a better way of doing this. We needed a way of testing all of these differences simultaneously in such a way that the chances of making a wrong decision are really low. And that's what an analysis of variance does. So the union of events is the sum of their individual probabilities, which is how we got to 0.5 from a bunch of 0.05s. So we have these means, and we want to know if any of them are different. So we already know how to calculate within sample variation, but we want to know if this mean is different from that mean, if this mean is different from that mean, etc. But there's another type of variance that we can calculate. I am totally not on my game today. Let's skip this. I'll come back to this. Whoop, where am I at? We can calculate the grand mean. So we can throw all of these things into a pile and say, what if treatment doesn't matter? We're going to calculate the grand mean. And when we calculate the grand mean, we can see how different the means of the treatments are by comparing them to the grand mean. So what you would do is you would say, how far is this mean from the grand mean? How far is this mean from the grand mean? How far is this mean from the grand mean? How far is this mean from the grand mean? And how far is the yellow mean from the grand mean? And we would do the same thing here, except that in this case, instead of the sums of squares with the individual values, it's the sums of squares of each of the means of the treatments minus the grand mean squared. That tells us how far the means vary from a set marker in space, which is the grand mean. This is how we would calculate a sums of squares, but in this case, it's the sums of squares due to treatment as opposed to the sums of squares within samples. Okay? It's a sum of the squared deviations, but in this case, the squared deviations of the sample means from the grand mean squared. And that gives us two kinds of variation. In this experiment, we have variation among means, we have variation within means, but there's another type of variation. There's also total variation. Sum squares total. How might we calculate sum squares total? Based on what we've done with sum squares within and sum squares among treatments. Remember, sums of squares within is the mean versus each of the individual values within the sample. Sum of squares among is the difference between the grand mean and the sample mean. If you want to know what the total variation is, what would that be like? Yeah, Sonia.
The grand mean and each individual.
Yep, that is exactly right. So you can quantify the total amount of variation in this data set by asking, to what degree is this grand mean different from this data point, different from these three data points, different from that data point, different from that data point, different from that data point, different from those two data points, different from those two data points, and different from this data point, different from that one, different from these three, different from those two, different from that one, different from those two, and different from those two, different from that one, different from those three, different from those two, different from those two. And, do I need to continue going on? You do that for every treatment. All of that squared, the individual values minus the grand mean squared and summed up, that's the total amount of variation that is in the entire data set. So one way to think about an analysis of variance is comparison of different types of variation. We have variation, that is the sums of the squared deviations of each observation from the grand mean. That is sum squares total. Comparison, the sums of the squared deviations of each observation from that observation sample mean. That's sum squares within, and whatever is left is sum squares among. If you know two of these, you can figure out the third. So if you know sum squares within and sum squares among, you can figure out sum squares total because they should add up to be that. If you know total sums of squares and sum squares among, you should be able to figure out what sums of squares within is. If you're working this by hand, one of the easiest ways of finding sum squares within is to calculate sum squares among. In order to find total sums of squares, which requires, in this case, 50 observations, 50 calculations, the thing to do is find within sample sums of squares, among sample sums of squares, add those two things together, and it will give you this one. If you're just starting out, the way to do it is calculate all three and verify that all of your calculations worked. So what matters is how this variance is partitioned. There's some variation that is due to the means of the treatments being different. There's some variation just due to random variation within the treatments, and those two things add up to be the total amount of variation. So, this is what it looks like. But do not let this frighten you, and do not memorize this. This is just quantifying the different things that you would calculate. This is the sums of squares within. It's the individual values minus the mean squared. This is sum squares within for this particular treatment. Sum of squares within because it's once again the individual values minus the mean. Sum squares within, sum squares within, sum squares within. All of those things added up across all of the treatments would be sum squares within. It's just the sum across all individuals within treatments and all treatments of the values minus the mean squared. Sum squares among, though, takes the means, compares them to the grand mean, squares those sum deviations, the sum square of the square deviations between the means and the grand mean, that squared multiplied times the sample size. In this case, the sample size is 10. That ends up being the amount of variation among the means. And then this is just what we did before. Sum squares within plus sum squares among is sum squares total. They're not showing you the calculation for sum squares total because it's just a pain to calculate. Because you have to do the grand mean minus each individual variable. And you can do that, but it's not necessary, assuming that you've calculated sum squares among and sum squares within correctly. So, yeah.
So, as Rick Lucy just, as Paul just said, you don't have to memorize these equations, but again, you know how to read them, and that's why it's still the same here. What can remind me what the little subscript I means in these equations? I'm going to come up a few times in the socks. What's the little I mean when you see it after an X variable?
In this case, it's really easy because you've got it at the top. I1, I2, I3, I4.
Not, well, what part of the data set is represented by these four columns?
Is it columns or rows? Columns. Is that what you're looking for?
So you're indexing through the columns. That's where the I usually comes from, because we're indexing through some observations, in this case, the columns. Notice that suddenly, since we're in a matrix, there's now also a little J showing up. What do you guys think the J represents? The rows. The observations within each.
Yeah. So if you're doing sums of squares within, it's the first column, first row, minus the mean of that column. So first column, first row. So within, you sum within a column across all the rows, and then you go from column to column to column.
So these are just telling you, which number, which actual value that you've got in your real data set.
Yeah.
To use.
And once again, easier than that is recognizing the different forms of variance that you have. Variance between the individual data set values, data point values, and their sample means across all samples. That's within sample variance. Differences between the grand mean and the sample means, that's among sample variance. And then you don't have to remember X's and I's. You just know kind of what you're doing. When you get this, all this information, you'll see a table. Or you'll create a table, actually. And the table will have the sums of squares, whatever the answer is when you've calculated that. Sums of squares among the treatments, sums of squares within the treatments. But then there's also a column here for degrees of freedom. Remember that when you calculated the sample variance, you calculated a sum of square. Then what did you do? You divided that sum of square by what? What is n minus 1? It's the degrees of freedom. It's it's a measure kind of how big your data set is. The bigger your data set is, the less these deviations from the mean matter because you have a really good idea of what the mean is in this case. So in all of these things, you're going to take a sums of square and you're going to divide it by the degrees of freedom. So sums of squares within is divided by the sums of squares, sums of squares within is divided by the degrees of freedom associated with within sample variation. And it's simply the sum of all of the data set minus the number of treatments that you have. So in this case, we had 50 samples spread across 1, 2, 3, 4, 5 treatments. So each treatment has 10 observations. So this is 50 minus 5, so that should be 45. The degrees of freedom for the treatments, among treatments, is the number of treatments, which is 5 minus 1, which should be 4. So this would be the sums of squares among divided by 4, and whatever the sums of squares within is divided by 45. These things are called mean squares. Because it's just the sums of squares divided by some measure of confidence, measure of sample size. You have 4 treatments, you have 50 observations, and this gives you a amount of variation that is scaled by the number of treatments you have. Obviously, the more treatments you have, the more chance for variation you have, simply because you're adding up variation across a greater number of treatments. Same thing with the sums of squares within. The more observations you have, the more you're going to rack up variation, so you divide that by the number of samples that you have to correct for differences in sample size. And so these are your measures of variation, either the sums of squares or the mean square. But when we go to make a decision, we actually look at how big is the variation due to treatment. This is variation due to treatment differences. Divided by the amount of variation that is just random noise. Random variation within your data set. Imagine that you have 2 means, and these 2 means have a lot of underlying associated variation. If you were asking the question, are these 2 means different, would you say that these 2 means are likely to be different or likely to be not different? All in favor of different, raise your hands. All in favor of not different, raise your hands. All unwilling to commit because you're afraid of being embarrassed, raise your hand. Oh, come on, that's the rest of you. Think about it in terms of 95% confidence intervals. Is the 95% confidence interval on this mean going to be really wide or really narrow? It's going to be really wide. If it's really wide, is it going to include the other mean? So would they be different or the same? They would be the same. Take this same situation, same distance between the mean of sample 1 and the mean of sample 2, but there's very little variation around those means. Would you now conclude that those means are different or the same? So what matters is not only how far the means are apart, but also how much within sample variation there is. Those means are exactly the same distance apart. All I changed was how variable the underlying data were. And that changed your answer. So to know if means are different, you have to look at how far apart the means are, but you also have to look at how much variation there is within the samples. Within sample variation can obscure differences between means. That's what's happening here. This variation within this sample is obscuring the fact that these two means may actually be different. They're not the same, but what prevents you from saying that they're different is there's too much variation within each sample for you to be able to see it. Okay? So we calculate this thing called the F ratio. And this tells us whether or not things are different or not. And you can look up an F ratio in a table, and it will tell you whether that F ratio is significant at the level that you're testing at. So for this data set, and now I've marked all over my screen, which is what I intended to do. So you can plug these data into a spreadsheet, which is exactly what I did. I did this in Excel. And you can calculate the sum of the values is 70.1. You divide that by the sample size to get the mean of 70, the sum is 701. Divide that by 10 to give you a mean of 70.1. The sum of the squares within is just 75 minus 70.1 squared plus 67 minus 70.1 squared plus 70 minus 70.1 squared, on and on and on, add it all up. Some squares within, some squares within, some squares within, some squares within, some squares within. That thing all added up equals up to 245. Sum of the squares among is this mean minus that value squared. So let's look at that. 70.10 minus 61.94. This is going to be complicated now because life is complicated. That's 1. That's 8. 8.16. When we square that, that should be 66.59. 59.3 minus 61.94 subtracted and squared should be that. But what's 66.59, 6.97, 13, 15, and a 4? Does that all add up to 1,077? Why not? We forgot to do something, didn't we? Nope, we squared it already. You have to weight those by what? Sample size, yep. So 66 and 6, that's 72, and 13 is 85, and 15 is 100, and 4 is 104, and then 59 and 97, 16, 9, 10, 15, and then 99, and then 52, and then 67, so that's 104. I added the whole numbers, and now I'm adding the decimals, because that's just how my brain works. Don't try and figure it out, just roll with me. .68. Oh, so I'm doing this in Excel, so it's carrying out many more decimal places than I'm carrying out in my rough calculations. But if we take this, what's the sample size for each of these treatments? Multiply it times 10, we get that answer. So you have to weight it by sample size. If you're curious, when we go back here, remember that you take these differences and you multiply them times the sample size. So that's why that looks bigger than it actually looks over here is because to calculate sum of squares among, you weight it by the sample size. Sorry, I do math maybe differently than you guys do, but so we now have sums of squares within, we have sums of squares among, that plus that should equal sums of squares total. Sum squares within, sum squares among, sum squares within, total. Here's your degrees of freedom. That divided by 4 is 269. That divided by 45 is 5.46. You divide that by that, you get an F statistic that is gigantic. This is an F statistic that is huge. Whenever you have a big F statistic, you're more likely to be signaling that things are different. It doesn't tell you what in the data set is different from what. It just tells you that somewhere in there, something's different from something else.
At least one treatment.
At least two treatments are different from one another. It doesn't tell you which two. Could be any of them. Now, we already have a hunch, right? You already have a hunch of what's different from what, right? My guess is that the control is different from everybody else. My guess is sucrose is different from all of those, and sucrose is different from these. And what do you guess about these? They're not different. But because you have two big differences, you have big difference between here and there, you have another big difference between here and each of these, and you have a bunch of differences between here and each of these. That's why your F statistic is so gigantic, is you have lots of differences in this data set. The fewer differences you have, the smaller that F statistic is. So we get to this answer down here. This is the F ratio. The ratio of how much variation is there due to treatment divided by how much variation there is just random floating around variation that doesn't mean anything within sample variance, among sample variance. That gives us that F ratio, and this F ratio gives us a p-value. And when you calculate P and R, the smallest value of P that R will ever produce for you is 2 times 10 to the minus 16th. If you get to the low end of the p-value, that's just it. So somebody remind me, what is alpha? What's that? Okay, so oftentimes we set it at 0.05, but define it for me. Okay. It's the type 1 error rate. It's still on the sideboard there. Sadie, what you said is actually correct also, but this is the name that we would associate with it. Type 1 error rate, it's the probability of concluding at least two means are different when there are no differences for this data set. Okay? So we set that. So alpha is something that you set, you the experimenter set. And when we say that we have an alpha rate of 0.05, a type 1 error rate of 0.05, what we're basically setting up is we're setting up a criterion for either rejecting the hypothesis that means are different versus accepting that hypothesis that means are different. And what we're saying is we only want to make a type 1 error 5% of the time on average. If we did lots of experiments, on average, we would make a mistake of saying that means are different when they're not 5% of the time. If this is what alpha is, then what is this thing called P? Do any of you guys know that? Any guys been exposed to P in the past? P is the observed type 1 error rate. So for this data set, if you say that means are different, you might be wrong, but rather than being wrong 5% of the time, you're going to be wrong this percent of the time. It's the type 1 error rate that is due to the patterns of differences in your data set. And so when you have a type 1 error rate that is this tiny, it means that when you say that there are differences in there somewhere, there are differences in there somewhere. The odds of you saying that there are differences in there and being wrong are astronomically low. So that's another way in which you can make a decision about your data set. Once again, it doesn't tell you which means are different from which other means. It just tells you that somewhere in there, differences occur. And there are other methods for figuring out what's different from what, which we're not going to go through today. P is what comes out of your data in terms of type 1 error rate. Alpha is what you decide is the criterion for making a decision. If you choose an alpha of 0.05, then your p-value has to be below 0.05 for you to say that differences occur. If it's above 0.05, you conclude that they're the same. Being above 0.05 is when you cross one of these two boundaries. When you cross this boundary or you cross this boundary. That's when you go from being below 0.05 to above 0.05. It's crossing that line. But it's an arbitrary line. Who sets what your type 1 error rate is? Speak up, I can't hear you. You do get to choose. You do get to choose. Oftentimes we choose 0.05 because it's convention, but you could choose 0.01, which means you only make a type 1 error 1% of the time. You could choose an alpha of 0.1, which means that you'll make a type 1 error 10% of the time. So what does that mean in practical terms? So imagine you are an ecologist and you're doing a big experiment, and because you're doing a big experiment, you can only have 3 replicates, really small sample size. So what that means is that you might want to see differences that are hard to see because you only have 3 sample sizes, which means that your within sample variation has the potential to be very large because you only have 3 replicates. But nobody's going to die if you make a wrong decision. And so you might say, well, I want to see differences if they exist. So I'm okay with saying that 2 treatments in my experiment are different and making a mistake 10% of the time. Now imagine that you're a doctor, you have a cure for cancer, and the cure for cancer you're pretty sure works. The bad thing is it's really toxic and it fries your liver. So at the end of your treatment, you're cancer-free, but now you need a liver transplant. Do you want to say your drug works and make a type 1 error rate 10% of the time? Do you want to make that error 5% of the time? No, you might want to make that error less often, and so you might choose a type 1 error rate to test against that is more stringent. Well, the tricky thing about that is that in order for you to see a difference at an alpha of 0.01, your differences between the treatment and the control have to be much bigger in order for you to see that treatment. So when you do see that treatment effect, you're pretty sure it's true, but what that means also is that it might be effective and you might miss it because you're asking the test to only tell you that it works when it really, really works. So you can avoid making a type 1 error, and you can be more and more sure that the differences that you see are actually true. But as you do that, you risk making another type of error called a type 2 error that is saying that two things are the same when in actuality they are different. As you run away from one type of error in statistics, you run headlong into this other type of error in statistics. We'll pick up there on Wednesday. We got a little bit more to say about this. Two things. There's a homework assignment that is a practice homework. It's a data set that is an ANOVA data set that is designed to get you to work through the equations so that you know what the mechanics are of ANOVA being done. Second assignment is a preparation assignment. It's a Google Earth-based assignment where we ask you to go to Konza Prairie on Google Earth and look at Konza Prairie and make observations about Konza Prairie, because on Wednesday we're going to start talking about Konza as an ecosystem that we'll be studying for the next 3 weeks. So that's your homework. Both of those are on Moodle, so you should be able to just download them from Moodle.